Great. Lovely. So everyone, welcome to our webinar session on Stony coral tissue loss disease. I am Khadija Stewart, Caribbean Regional Representative for SOA and the Hub Leader of SOA Caribbean. And co-hosting this session with me is Prophet Tiburon. Carlos, go ahead and introduce yourself.
Hey guys, well, I am Prophet Tiburon, Carlos Perez Murcia, and I am the president for SOA Colombia, and I'm a scuba diving instructor. So everybody guys from Colombia, let's meet in SOA Colombia and let's do some ocean conservation and restoration and medication, because now we need medicine for corals. So Kali, we do some ice breaking before we start with these amazing speakers that we have today that are right now action on saving the corals.
Let's do it.
We try this ice breaking activity and we start.
Right, so it's just a poll, guys. I don't worry, don't feel like you have to put on your cameras and do anything crazy. It's just a poll. So this is the first time I'm doing one of these. So if it doesn't work, let's just pretend that it wasn't part of the plan and we would just jump straight into the session. Okay, so let me start and go ahead. Is everyone seeing it on their screens, or am I failing?
Yes, I can see it.
Okay, lovely. All right, we have some answers coming in. We had 60% participated. Come on, guys. Don't leave our poll hanging.
Profe, if you're speaking, you're on mute.
Oh my God, I was speaking a lot. Okay. So let's discuss a little bit the questions. I see, what is the cause of stony coral tissue loss disease? Virus have some boats, virus like COVID. Bacteria is winning, 60%. Heat, that's an important one. Heat has some answers. How many species does it affect? 24 to 30 species is winning, 56%. 0 to 5 species has some votes. 10 to 15 species, 32%. Is a stony coral tissue loss disease present in your country's water? Yes, no. Has your country taken action? Yes, 32%. No, 20%. I have no clue. 50%. So that's important because we don't know what our countries are doing. And we have 85% of the people here that has the sickness. So Kadi, awesome.
All right, well, thank you guys for taking part in the poll. Let's kick starting. So we have our first speaker, Hannah Lochan, who is from Trinidad and Tobago, and she's representing SOA Caribbean, and she's going to be talking about coral reefs. So Hannah, go ahead, introduce yourself, and we are all ready for your amazing presentation. You have this, don't worry. Floor is yours.
All right, thank you so much, Khadija. Hi guys, my name is Hannah. I am from Trinidad and Tobago. I'm a marine scientist and I'm also an MSc student. So today I'm just going to be doing a brief introduction to coral reefs for you before these amazing ladies take it away with the stony coral tissue loss disease. So I'm just going to share my screen. Share. Right. Okay, great. So y'all can see my screen, just confirming.
Yes.
Great.
Yes.
Okay. So hi. Like I said, we're gonna just be introducing coral reefs, and I'm gonna do it from the angle of Tobago, where a lot of my work is done. So firstly, we're just gonna look at what is a coral. So some people are like, a coral is a rock, it doesn't move. Or some people would tell you it's a plant, it has these things and it looks like a plant, but no, a coral is actually an animal. So a coral eats, it reproduces, it grows, it respires, and this hair is just the basic anatomy of a coral. So you have the tentacles, they have little nematocysts inside them, and these are the stinging cells. So some corals actually sting harder than others. If you've ever gotten stung from a fire coral, you know what I'm talking about. And then the internal section is more where the tissues will be responsible for reproduction, digestion, and feeding. Also within the coral, there is tiny little microscopic algae called zooxanthellae. So these have a mutualistic relationship with the coral. So the coral respires and produces carbon dioxide, and then the little algae within them, the zooxanthellae, will use this for photosynthesis. So it's a nice little cycle, and then the zooxanthellae will produce food for the coral. So corals mainly get their food from their zooxanthellae, which is about 90% of their food budget. Also, and then on the right hand side, we have a nice little macro photo of Acropora palmata. So you can't see the internal structure, but at least you can see the little tentacles that I think look quite cute. So where are coral reefs found around the world? So corals are quite picky about what conditions they like. They like warm water. They want nutrient poor water. They want a high salinity. So this allows them to flourish in the zone along the equator. So we usually find corals within along the equator and within 30 degrees north to 30 degrees south of the equator, as you can see in this map from the Allen Coral Atlas. So on this map, all the red little dots that you're seeing all over the world, from South America, go up around Africa, around Indonesia and Australia, all these are coral reefs. So now I'm going to take you to Trinidad and Tobago. And I don't know how many of you, if all of you know where we are located. We are actually right at the southernmost of the Caribbean archipelago, and we are quite close to Venezuela, about 12 to 20 miles away from Venezuela. So Trinidad and Tobago is very special because our waters are really nutrient rich, because we get a lot of the runoff from the Orinoco rivers and the Amazon rivers from South America. So our conditions are a bit environmentally different as compared to islands in the Northern Antilles. So we do have, our coral reefs are pretty special to us. And this map here just shows the location of the coral reefs around Tobago. So we have right here in the southwest, we have the Buku Reef Park, and that is the only marine protected area that we have in Trinidad and Tobago. And it's quite beautiful. And then we have coral reefs extending along the Caribbean coast, and then also in the Atlantic Ocean. So everybody loves coral reefs. We know they're very important. I'm just going to discuss a few benefits of coral reefs. So this image is showing Buku Reef, an aerial image of Buku Reef. And as you can see, the reef extends pretty far out. It's about 7 kilometers squared in size. And what's very important is that you can see that in the picture, waves are breaking not on the coastline, but they're breaking on the reef. So coral reefs provide a really huge structural framework that allows the dissipation of waves. So waves break on the reefs, and so instead of breaking on our coastline. And this is for Tobago a multi-million-dollar resource from these natural resources. Also, South Tobago is pretty low-lying, it's flat. So these communities would be very vulnerable to coastal erosion if it is that we did not have this huge coral reef system along the coast. Secondly, major benefits of coral reefs is tourism and the livelihoods that benefit from them. So livelihoods can be benefited directly and indirectly. So we have boat tour operators. Here we see a glass bottom boat, which is very popular in Buku. People will pay money to go out on the boats and just spend time in the reef and in the nylon pool. And then also directly impacted would be fisher folk, boat tour operators, dive shop owners, and then indirectly would also be the whole hospitality institute that is in Tobago that is there to accommodate visiting tourists. And this contributes directly to our economy and helps add some sort of diversity to the economy. Thirdly, we have biodiversity and life. We are able to get food from our coral reefs, and that is so important. And having the more fish we have, the more life we have, the more we will be able to benefit. And here we have a picture of a pretty little parrotfish. And as we know, parrotfish are grazers. They eat algae on the reef, but also when they graze on the reef, they also sort of bite at the coral. And for those who didn't know, parrotfish poop kind of turns into sand. So all the white sand beaches we have in the Caribbean and by coral reefs, a large part of it is due to these parrotfish, making them so important for coral reefs. Also, we just have, the aesthetic beauty of the coral reefs, of the coastline, and this is also very important for society to have this a free recreational resource. And the sea is symbolic often of relaxation and reflection. And they say healthy reefs equal a healthy society, and it really is very true. Now we discuss all the benefits. As we know, corals are highly threatened, very susceptible, vulnerable ecosystems, and there are quite a few threats that they face. So one such threat is coastal runoff. So when we have development on islands or in places and it's not properly managed, you might have coastal runoff of sediment or of pollution, and having this in the water can smother the corals. It can change the water quality so much that the corals can no longer grow in that area. And also sediment mixed with wave action can actually scour the coral's tissues and damaging them, making them further susceptible to other stresses. So in this picture, you can kind of see, a few hundred meters off the coast, it's like brown water. And then you kind of see a line where you see the sea. So that sediment really is not good for coral reefs. Again, increased sedimentation and increased nutrient pollution kind of changes the benthic organisms that benefit from the quality of water. So you might have more turf algae and you might have more soft corals growing. And the problem with this is that these organisms don't provide as much structural integrity as a hard coral would. So when you have a reef that's full of turf algae and soft coral, it also competes with hard corals so that they no longer have this hard substrate on which they can grow. Also, soft corals encourage sand deposition. So they waft and then the sand kind of falls. And then the more sediments you have, the less space you're going to have for corals to grow. And here, so in Tobago, we actually have pretty low recruitment. And it's possibly due to high turf algae on the reef. So here we have a little Agaricea recruit. And it looks pretty good, but you can see that there is turf algae, there is macroalgae growing very close to it. And the growth rates of turf algae and corals, are very different. Corals grow so slowly, turf algae grows so quickly. So having high amounts of turf algae, it really reduces the potential for coral recovery and coral regeneration. Again, we have parrotfish. So there is a problem with overfishing, especially when you fish your herbivores. So when you fish your herbivores, you not only remove the prey for top predators, you also have less fish that are grazing the turf algae, which you need removed from the reef. And again, like I said, the parrotfish poop is beneficial as sand, and just the less parrotfish you have, the less healthy your reef system will be. We also have the invasive lionfish. I think there's been pretty good lionfish awareness over the years and throughout the Caribbean, and lionfish eat everything, and their stomachs can expand by 30%, and they have no known predator except us when we go spearfishing. So the potential for a lionfish to really deplete a coral reef is very high. Luckily for us, we have discovered that lionfish are absolutely delicious. So that's one plus for us, and we can actually spearfish them, and in a sustainable way that will also be protecting our reefs. Another threat to coral reefs is bleaching. So currently Tobago and the wider region is under a coral reef bleaching alert level 2. And when you have bleaching, the zooxanthellae that I previously mentioned that lives in the coral, they actually are expelled and they're lost. So when you have a coral that no longer has its zooxanthellae inside of it, it no longer is getting the food and the food source that it needs. So as you can see, this is a little, not little, it was pretty big, a Copophilian matans. So you can see the brown areas are where there's still zooxanthellae that's photosynthesizing, where parts of this coral are still alive. But where you have white areas, this is where zooxanthellae has been expelled. So with the zooxanthellae which provided the color for the coral is gone. So now the coral exoskeleton, which is white, is now exposed. And having less zooxanthellae, this means that the corals will be starving and they would be even more susceptible to disease. So as you can see here, this is a Siderastrea siderea. It has dark spot disease. It has gold mortality as well as bleaching. So, you know, it's kind of like during the pandemic when we had COVID and they would say, you know, healthy individuals will recover faster, but if you have comorbidities, you are more susceptible. That's kind of what it is. When you have bleaching or you have these other stresses on you, you're even more susceptible to further and further problems. Again, it's really important to note that there are some global issues. There are some huge issues like sea surface temperature increasing, global warming that we can't really tackle at a national level or at an island level. But there are some of these localized threats that, you know, mitigating these threats can still help our corals to build their resilience. So in Trinidad and Tobago, the key or agency that collects data on coral reefs is the Institute of Marine Affairs. And they have been collecting benthic data on reefs in Tobago for over 10 years. And when they look mostly at hard coral, soft coral, sponges, calcareous algae, and macro and turf algae. And what we can see here is that in 2010, the dark green line that you see here, this represents hard coral. So we can see in 2010, they actually monitored before the mass bleaching event that occurred within the Caribbean. And they got a pretty high coral cover. And after that mass bleaching event in 2010, coral cover declined. And it's pretty stable. Around Trinidad and Tobago, we have approximately 15 to 18% coral cover, which is not exceptionally low, but it's not that great either. And what is also interesting and important to note is the yellow line, which is turf algae. So in 2010, we had about 10% turf algae on the reef. And by 2016, it was about 40%. And when you think of just the surface, if it's just covered by algae, that still increases competition between turf algae and hard corals. And this is just to show you a few other sites. So it doesn't really change too much. All the sites pretty much a stable, a declining / stable hard coral cover, but with a very high turf algae cover. So building resilience in Tobago. This year, the Institute of Marine Affairs was able to conduct experiments looking at coral spawning monitoring from July to October, because we know recruitment is low. We wanted to know why. And you know, are our corals even spawning? And it was fantastic news to find out that yes, they are. So this is actually a mountainous star coral that was videoed in August. And you can see it releasing its bundles of egg and sperm. So that was really great news for us. And many would say not many, maybe a few other species also, we also got data for that spawned. And on the right hand side, this is just a macro photo. You can see the egg bundles popping out of the coral. So right now, Tobago has no record of stony coral tissue loss disease. We know it's not a matter of if we're going to get it, but more when we get it. So we monitor the reefs of Tobago every year, as well as there is the Institute of Marine Affairs has an app where they ask persons, if you observe something strange in Tobago, just send it into the app, send pictures, and they will check it out just to make sure, if it is stony coral tissue loss disease, we definitely begin and monitoring to determine the extent and prepare an action plan. So that's it from me. I'm just going to hand you over where you'll learn even more about stony coral tissue loss disease.
Thank you so much, Hannah, for that excellent presentation and really setting the foundation for the importance of corals. I can't speak about the importance of coral and really, setting that foundation so that now we understand the benefits, what they are, the threats that they're facing, and now we're really going to dive into stony coral tissue loss disease. Carlos?
Okay, super nice. Yes, Hannah, I think it was a good background of the threats, and we have a lot of threats, and now we have a bigger one, that is stony coral tissue loss disease. When you see it in your reefs, then you're gonna see like the magnitude. So let's talk about what's happening in Puerto Rico. Yes, Michelle Baptiste says, great job, Hannah, congratulations. We have Mario from Jamaica. So hello, Mario, welcome. Remember, guys, to do some stories on Instagram, tag us, SOA.Colombia, SOA Caribbean, Roatan Marine Park, Amando Oceano PR. And now we have Adriana del Mar Bayo and Lourdes Marcano, right from Puerto Rico. Puerto Rico is a beautiful place I want to know. And tell us, Adriana, how has it been with the sickness? Is it new? How are you guys treating it? So the floor is yours, and thank you for saving the Caribbean reefs.
Hello, everyone. My name is Aliana del Mar Bayo, and I am the Marine Coordinator of Activities in Amando Oceano. And today I will be talking a little bit of what is SCTLD. And SCTLD, as what it says in the short letter word, is stony coral tissue loss disease. And this will indicate that the coral does experience tissue loss and is then with an exposed skeleton, as we see in the picture right in the right hand. This is this is a disease that has an unknown pathogen and an unknown cause, and it affects more than 30 species of hard corals. It was first identified in Florida in 2014, but has since had a spread throughout all of the Caribbean, and you can also see various symptoms by firsthand. First is the multiple lesions that are formed around the infected coral, and eventually we can see the complete colony being engulfed by this disease. Then we also have that there is a very high mortality rate that is also due to the fact that it is very contagious and it spreads very quickly. In the next slide, then we see the story of the disease. As I mentioned, it was first seen in 2014 in Florida near Virginia Key. Then it starts going down, spreading throughout the southeastern Florida coast, then towards the Florida Keys, where it is suspected in Jamaica, but it has not been reported yet. And then towards the 2018, we see it in San Martin and the Mexican Caribbean. So we now see that there is a very rapid spread along the years. Then in 2019, we see that it is in beyond Key West and St. Thomas and the USVI. Later on in 2019, we already see that the Dominican Republic, Turks and Caicos, Belize, and other islands are being affected quickly. Later on in 2019, we see that it finally got to Puerto Rico and Culebra, And most recently in 2020, it was then reported in St. Croix and New Providence, Bahamas, Guadalupe, and St. Lucia. So as we can see, it is quickly going all around the Caribbean and affecting large quantities of area. In the next slide, we now see that there are the susceptible species, and these susceptible species are they have various characteristics, and one of them being that they are slow growing and long-lived reef building corals. And among them are the smooth flower corals, the pillar, elliptical start, maze, boulder, grooved, knobby brain, and symmetrical coral. All of these, which have different levels of susceptibility, still pose a huge victims, as the threat of SCTLD is affecting a very large quantity of species. Now we see that in Puerto Rico, as I mentioned, it arrived in 2019, but it was on the island of Culebra. Since then, it's been reported in more than 80 places island-wide, as published in the DRNA interactive map, which I will show soon. And since then, there has been more than 20 localizations that have been referred to as the treatment centers for the disease. The treatment we are currently using is a mixture of amoxicillin and base 2B, and this mixture is applied directly onto the coral, where it sometimes will last between 1 to 2 weeks, and it will usually have to reapply. So this brings into the challenges that bring us to the application of the treatment. Then it would become first apparent that there is a lot of rip currents in Puerto Rico, and a lot of chances that the treatment will not stay on the coral. Other than that, there is very limited amount of treatment and limited amount of personnel. So when an area is being treated, we usually have to pick which are the most severe corals, and then treat those and leave other corals that are still being affected untreated because there is not enough treatment. And this could also pose a threat because it is a very contagious disease. So we still have a contaminant in the area of treatment. Here we see in the interactive map by the DRNA, which is the Department of Natural Resources of Puerto Rico, we see that the 80 places with the spots that are red are the places where the incidence has been reported. And the stars with the yellow dots are the places that are being treated. As we see, it started first in Culebra, but then it started going south and then southwest and then towards the north. And that is when it engulfed the island in a matter of 3 years. So it is a very severe disease. Here we see then a map of where are the reefs localized on the island. We see that in Culebra, there is the largest quantity of reef covering that area. And it could be apparent that this is the reason why it was first seen the disease there, and then that's why it started spreading towards the south, and then the southwest, and then the north. So it started where there was more coverage of reef to where there was less coverage of reef. And this could also give us an insight to how it will keep on spreading around other places where there is the conditions for this disease.
Hello, I am Lula Marcano. I am a program coordinator for Amando Oceano, and I am an undergraduate student from University of Puerto Rico, Mayagüez. I'm gonna be continuing this presentation. So after about a year that we first saw the disease in Culebra, the government started to respond to the disease. So in June 8, 2021, there was an administrative order that was signed that detailed that SCTLD is established as a threat to the reefs of Puerto Rico, and a response began to be coordinated. Then, by August 30, 2021, there was an executive order that was signed declaring an ecological emergency caused by SCTLD in Puerto Rico and ordering the necessary measures to be taken with 1 million since we were, the disease spread so quickly, we needed to start acting against it. Although those $1 million still, we haven't seen them. Here we can see 2 pictures of corals that are being treated with the amoxicillin and 2B base. You can see the skeleton of the coral, that is the white part. The coral is not bleached. That part of the coral is already dead. Then on the left part is where the coral is still alive, and between it is the paste that was applied in Rincón, Tres Palmas, which is in the western side of the island. Here we can see how the disease progresses on the coral, which is very quick. You can see from the first picture to the third one, not even a month has passed that the coral is already almost completely dead. There is only a little tip that is still alive, but I'm sure that it quickly died off from this coral. So that is why we have to act very quickly before even knowing more facts about the disease itself, because of how fast it is affecting our corals. So what are some intervention strategies that are being used? There are different types of intervention strategies. First of all, there's monitoring of the corals. So if someone reports that there is a coral with SCTLD, that coral is then monitored and the surrounding area, because usually it starts spreading. There's also treatment. So they are mostly using amoxicillin, I have the understanding, which is the antibacterial paste. And there are also some probiotics which are used. Other ways to intervene with the disease are coral colony amputations and just whole infected colony removals, and minimizing environmental stressors to corals, since, well, the coral is sick, and if those environmental stressors make it worse, it's like when a person is very stressed and then they get sick because of it, they cannot heal properly because they are animals. So what can a regular person do to help with this underwater pandemic? You can become a citizen scientist. You can take workshops to learn how to identify this disease, to report it. And the Atlantic and Gulf Rapid Reefs Assessment also lets you be kind of like a citizen scientist, where you can learn more about the disease and spread awareness. Another way that people can help is when they go either just normal diving or maybe to apply treatment. It is very important to keep the gear clean, to go from the sites that are healthiest to the ones that are more diseased instead of the other way around, and to properly dispose of and disinfect rinse waste into sink, tub, or shower. So they're just some of the ways to lower the risk of spreading this disease, since it spreads very, very quickly. So yes, that is our presentation, and I hope you could learn something about this disease.
Awesome. Thank you, Lourdes. Thank you, Adriana. I think you, Edgardo Bayo, super nice, super nice. Awesome child. Bueno, Adriana, I love the way you put, like, it's very serious and it's very rapid. And that picture, like a month, it was so difficult. It's good that you have started with the treatment and that you can continue, and that we found the money to do it, because as you said, the government gave the money, but where is it? And where in Colombia it's happening the same. Is one NGO getting some funding from people like in GoFundMe and putting the treatment, and the government is like, okay. So it's bad, and I think maybe like in Trinidad we should be ready before it gets, because it will be get there. So start looking for funding would be very important, since for Colombia and for Trinidad. Okay, thank you. So now we have, I think, the most experienced diver of Honduras talking about what's happening there, because as you see, the story of the disease, it went from Florida right to Mexico and right next to Guatemala and Honduras. So I think Honduras has a lot of years with the sickness. So I think in Roatan Marine Park, and Gabriela has a lot to tell us, and we have a lot to learn of the experience of Honduras. So Gabi, please present yourself, and the floor is yours. We don't hear
Sorry,
you.
sorry, I was muted. Thank you so much, Carlos, for that very generous compliment. I very much appreciate it. I'm going to start sharing my screen. Hope everybody can see it. I'm very happy to be here with all of you today. My name is Gabriela Ochoa. I am a research advisor for the Roatan Marine Park, one of the many organizations that have been working to mitigate this very devastating disease. And today I hope to share a bit more about the work that the Roatan Marine Park has been conducting in response to SCTLD. And Adriana did a wonderful job on sharing with us about the disease. And as she mentioned, this is a waterborne disease, and it's characterized by sloughing of the tissue. So basically this leaves a white intact skeleton. And there is a lot of debate on what causes the disease, whether it's bacterial, whether it's a combination of pathogens. There is still a lot to learn from this disease. But the latest science is telling us that this condition is caused by a disruption between the zooxanthellae, which Hannah mentioned is the symbiotic algae that lives in the coral, and the coral host itself. In Honduras, this disease, as Carlos mentioned, has been ongoing for quite some time. It was first detected in September 2020 on the southern coast of the country, and it's not, we expected it to happen, and we're not surprised that it happened in the southern coast, because that's where a lot of cruise ships come, a lot of cargo boats are coming, and we know that this disease is waterborne. It can move through water currents. It can also be transmitted through ballast water. So it's not surprising that it happened there. And it's very important, as Carlos was mentioning, to be prepared for this disease. And this is something that we did in Honduras. We actually started monitoring prior to the disease in January 2020. And we used a methodology called rover diver, where we are basically in the water, just tallying to see if we see any sick corals, any corals that have any symptoms or signs of concern. And this was an ongoing monitoring, which we conducted all the way to January 2020. And here I just want to show you the latest map that we had from January. I'm sure this has changed. It's been a while since we've done this type of monitoring, but you can see that at this point, all of the island of Roatan had SCTLD in what we call the endemic phase. This means that their prevalence of the disease is low. We're no longer seeing these highly susceptible corals. All that is left are resilient corals or corals with intermediate susceptibility. So there are no sites in the pre-invasion space, unfortunately. And a big part of our efforts were dedicated to intervention, which was treating corals with antibiotic and a special ointment designed to be absorbed by corals called base 2B. And this is mixed in a specific ratio, 400 grams of base 2B plus 50 grams of amoxicillin. This can yield about 6 syringes. And a big question that everybody asks us is, how many corals can we treat with that? Well, that really depends on the size of your corals, how far apart your corals are, your dive time. So in general, we can treat between 6 to 18 corals with one of these jars of base 2B. And currently, this is the most effective method being used to treat SCTLD. And we did not do this work alone. This required a lot of manpower, a lot of people in the field. We were lucky that we had many partners in the field, and we treated about 60 sites island-wide. This is just for the island of Roatan, where we are based. And our team actually spent about 600 hours underwater treating 47 sites. So I want to reiterate, sorry, that this is a very labor-intensive job. It requires a lot of work. So it's very important to prioritize, to have a very clear idea of what your goals are before you head into the field and start treating everything. Because as Adriana was mentioning, we can't treat everything. We have limited funds, limited people, limited resources. Overall, we've done about 3,000 treatments, and we've fate tracked about 1,400 colonies in the island of Roatan. And here, I just want to show you this group brain coral, Diplora labriformis, that we've treated in February. We reassessed in March. We thought it looked pretty healthy, maybe some disease happening or starting to happen here. We had to retreat in April, and then in May, it was looking a little bit better. And the reason I show you this slide is just to reiterate that we can't just treat corals and assume that they're going to be okay. There has to be a level of reassessment. We have to go back to that coral. The treatment stops the lesion, but it doesn't keep the coral from getting new lesions. So that's something to keep in mind when we're doing this work. And a big part of our work has been community outreach, sharing with people about this disease, just as we're doing right now in this webinar. We've had about 7 workshops to teach people about coral identification, because prior to the disease, there was very limited experience and capacity on coral identification, disease identification. We've done various trainings to teach people how to apply the antibiotic. So overall, we've trained about 303 people. We had about 30 volunteers help us with this work. And, we know that the disease is spreading. At this moment, there's about 25 countries or territories that have the disease. And especially in Honduras, we knew that it was going to move to different islands, to the coast. So we knew it was important to train other MPA managers and other organizations on what to do when they got the disease, so that they could be even more prepared than we were. Another program that we had was an Adopt Our Dive Site program, where we were actually working together with dive operators to have them adopt dive sites so that we could have more people in the water, we could have more hands helping. This has been a very gratifying project, because about 9 dive operators have joined our efforts and have started treating dive sites. We've also collaborated with the Gulf Coast Fisheries Institute to create posters specifically aimed at divers, because we know that it can be transmitted through divers moving from sites that have the disease to sites that don't have the disease. So we created posters to help stop the spread, teach people how to decontaminate, using different guidelines on how to breach the different types of gear that they might be using in the water. We also developed a citizen science data collection project, because we were putting tags on these corals, and Roatan is an island that many people visit for recreational diving. We created these little posters where people could take a picture of the coral and use this little QR code to send us the photos and help us reassess corals without having to go into the water. So trying to maximize the people in the water helping us. And very important, we also connected with the international community. We gave a lot of interviews to spread and let people know what's happening underwater. And we were all suffering a pandemic above land, but underwater, there was a very serious pandemic, and there's still a very serious ongoing pandemic. And so we shared our story in different outlets, including the New York Times. That was a big one for us. And, we talked about research, we talked about communications, but management is a very important part of SCTLD, getting governments on board. We all live in developing countries and Caribbean nations, and we all know that governments can always be the most helpful, but it's always important to bring them to the table. So as Roatan Marine Park, we've supported the draft of the National Action Plan, which is now being validated by different partners and authorities, and we're hoping that it will be completed soon. And as the disease kept spreading and we were reaching the endemic phase where there were very few corals to treat, whatever we treated was there, whatever couldn't get saved wasn't there anymore, we started thinking in the future and building capacities for coral restoration. So we visited different rescue centers to learn about how to rescue corals and bring them into land facilities. We also learned from colleagues in Dominican Republic about assisted sexual fertilization and coral spawning and things that we could do to rebuild our reefs after this devastating disease. And in late 2021, we began also our coral spawning monitoring to validate our spawning calendar. And this was in part thanks to training that we received from colleagues in Mexico and Sea Corps. And our big dream that we have is to be able to have Honduras's first coral restoration center, where we will be able to bring some of these surviving corals, but also conduct spawning work in the lab. And so just to end, I want to give those that are experiencing the disease that don't have it yet a few recommendations from people in the field. And one that's very important, I think it's important to establish intervention plans according to your conservation goals and your budget. We know that money is limited, so think very careful of what you want to save, because unfortunately we can't save everything. Carry out monitoring in very strategic places. Think about, if you don't have the disease, where is the best place that you think it would happen, right? So close to ports, close to cruise terminals, etc. Monitor corals that have been treated with antibiotic. Very important to always go back and check on those corals. And we learned that it's also very important to make maps of these corals, because navigating underwater and finding these corals intact can be quite difficult. Also manage expectations from your stakeholders. People get very scared when they learn that there's an underwater pandemic, and they want us to do as much as we can, but we're limited by funds, we're limited by time. So be very honest and transparent with your stakeholders and manage the expectations. And very important for your team, prioritize your mental health and that of your team, because this is a very devastating disease, and it's very tough on people's mental health. So prioritize that. And I want to thank everybody that's made this happen. And thank you all for listening. Here's our contact information, and I'll leave my email if anybody has any further questions. And thank you so much.
Thank you so much, Gabby. Wow, that was amazing. I feel like all our speakers were absolutely amazing. I learned so much. I don't know about you guys. I hope you did, but I know for myself, I definitely took away a lot of information, you know, the importance of one, getting government involved, having the community involved, community citizens-based science, and all these different elements. And just really understanding that, hey, this thing is happening and we need to take action. And even if you aren't seeing it as yet, you still need to take action. So thank you guys so much for coming and presenting. Carlos, any words, any thoughts?
No, that was amazing, Gabby. That was like the best way to end up this. And I feel you when you say mental health, because when I saw it, it's hard, and it's like, oh, what can we do? And then we don't have funding, and where's the government? And but we keep strong, and we have you ladies doing amazing work. So let's try to replicate that. We have some questions on the chat. There's something about the training. Gabby, you said something about training. So Kadi, Diana, I don't know. We have to do something for training.
Yeah, I mean, go ahead.
I didn't mention this in the presentation, but in the Roatan Marine Park YouTube channel, we have all the coral ID presentations that you can look at, how to identify SCTLD. So all of these trainings that we've given, because we had to experience SCTLD during COVID. So everything is online. So you can actually go on the YouTubes, and I'll put it on the chat box. For anybody that wants to learn more, they can learn there too.
That's
yeah,
awesome.
and the idea. I'm sorry, I'm gonna speak. The idea too is to get some Latin America and Caribbean youth present in some of these places, so Roatan and Puerto Rico as well, to learn firsthand how to deal with it, how to treat, how to see these things. I am forever looking for grants and funding and ways to come up with different, you know, projects and activities. And I think that would be something that hopefully a lot of you all here present would be interested in. I know I for sure I'm definitely interested in going and seeing firsthand and learning how to identify and treat. I probably have to learn to dive first. This is where Carlos will come in. He will certify all of us, and then we will get there. But if you have any questions, you know, you could unmute yourself. But before, I know, Carlos, you were saying something, so go ahead and speak.
Yeah, no, we have some other questions in the chat, but yeah, let's unmute. Let's get our camera on. We have the YouTube training. So Carlita, thank you for that questions. We have more questions right here. Honesty, or he or she asked, but can the corals be pre-treated? Can the corals be pre-treated? Asking for islands that have not yet faced this disease as yet. So maybe, I don't know who wants to answer this.
So there's still a lot of people developing different ways to treat corals, and there are certain scientists that have been looking at probiotics, that those can be given to corals before the disease to make them resilient. However, this has only been developed for certain species, and I haven't really seen much rollout of that. I think that what you can do if you don't have the disease is start working with dive operators to inform them about decontamination procedures. If people are coming from islands that have SCTLD to visit, for example, Trinidad and Tobago, make sure that the gear is disinfected or that dive operators are only using gear from that area. So that's my recommendation.
Yeah, and me, I recommend to look for funding. Like, because very, very difficult, and then when it gets right there, like Lourdes, you showed us in 1 month the coral is gone. So if you don't have the funding before it gets, well, it will be difficult. So get funding and training. There's one question for training, for this training. Maybe this could be like the first start of this treatment. How do you distinguish it from other diseases or just bleaching? And it's not that easy, right? Like distinguishing for bleaching and for stony coral tissue loss disease. How do you do it? And so maybe
Adiana, I feel like one of you
Diana?
guys want to take it, not to
I
put you
was
on the spot.
I was waiting for somebody else to speak, but I wanted to say that it is like a concentric lesion, and then it starts like spreading out. And when it's SCTLD, and when it is the coral bleaching, to my understanding, it is more like spotted. So it is like different individuals inside of the colony that are experiencing the bleaching because of the Zooxanthera leaving the area where they inhabit. But when it's a SCTLD, since it is large chunks of muscle depleting, you can actually see the skeleton as well, and it involves the colony completely, just like concentrically. So I don't know if that answers the question.
Yeah, and I just wanted to add to Adriana's answer that the way I love to explain it to people, it's like bleaching is like, you know, maybe you haven't been in the Caribbean for a while, you're starting to look a bit pale, you need to get a bit tanner, right? So you're kind of losing that Zooxanthella inside you, but you're still alive. You know, you're just a bit pale. But SCTLD is as almost if it's the flesh is coming out of your bones and leaving a white bone. And I think another way to know that it's SCTLD is Adriana was mentioning really well about the susceptibility and which species get it first. So for example, you would see pillar corals and mace corals showing multiple lesions, and you wouldn't just see 1 or 2, you would see 10 to 20 of the same species. So, you know, I always tell people, when you see it, you will know. It's kind of cliche, but you will know because it's something that you've never seen before.
Yes, and I've seen like some spots like chicken pox, you know, like and it starts like it's really like dead, then white, and then colored. So you have like the 3 like living coral, bleaching coral, and then dead coral like on the same area. So maybe that would be awesome if we have a training. There's people asking if there's training in Puerto Rico. Maybe Amando Oceano will give training later. I don't know. Lourdes or Adriana?
We have collaborated to make training accessible and have volunteers to put treatment specifically in Tres Palmas. And I also know that I think CESAM, which is an organization of marine science students. They also have professors that give training, and Islamar. I know that you can register to become a citizen scientist, and I think also receive training to put amoxicillin base onto the corals. So there are options. You can also contact Departamento de Recursos Naturales, who to my understanding, also give trainings to have more volunteers into the water.
Okay, excellent, excellent. Thank you, Lourdes. There's Javier saying it would be amazing to implement those projects in Panama. Awesome that we have people from Panama. I've seen some of the lessons while I'm diving, but I don't know well if there is any specific report. Thank you for spreading the word. Thank you for coming here. Thank you, Kyle. I was having difficulty telling the difference between bleaching and SCTLD. There's somebody asking if you know Daniela Larios. I don't know if she's like a coral. Yes, I see some yes. So I see Chandel. They know Daniela Larios. So maybe Chandel, if you want to talk something, you can unmute yourself. Is there any correlation between this disease and temperature changes in the water? No, temperature, no. It's
No,
more,
in fact, just to add to that, their scientists have determined that when corals bleach, prevalence of the disease is lower. So maybe bleaching is not such a bad thing for SCTLD, but when our corals are bleaching, there is less incidence of SCTLD, but there is no correlation between temperature and SCTLD.
wow, that's weird. And this is a nice question, maybe the last one. Yes. What is the
Yeah.
acceptance of the coral to the antibiotic? There is some coral that responds better to the treatment, perhaps the young coral or the coral in the mature stage. And I saw that the treatment, Gabi, in some pictures that you showed, it works. So yeah, what is the acceptance of the coral to the antibiotic? So maybe you, Gabi, that you have like the most...
Yeah, it really depends on the species. Some species respond more to others. I can speak only for Honduras because I think that also the rates of success really depend a lot on the country and the corals, but we are seeing something between 40 and 75% efficacy, not 100%. And this is at the lesion level, right? So this does not take into consideration that the antibiotic could work on the coral, but it might get new lesions, right? Because the antibiotic does not stop the coral from getting new lesions. It stops the lesions that are treated.
Okay. Well,
Hello, profe. Hello, profe Tiburón.
Edgardo, ¿cómo estás? Excelente, te felicito. Muy bien, cuéntamelo todo. Tell me
I like
everything.
to commend you guys. You guys are beautiful. That's we got hope in this generation with young guys, young scientists, female and males. We are really proud of having all of you, and especially having Adriana, pretty much taking over what once was my passion, which is scuba diving. Everything. And I'd like to add, besides commending you guys and felicitaciones, big heart, everything for everybody, love and kisses, hugs. I'd like to add that I feel like I'm at home. As a matter of fact, later in my life, I picked up this new hobby, this new project. Actually, I'm the secretary of the board of directors of Blue Tide Puerto Rico. And I think at the end is get a picture. And we are one of the most important projects that's being developed in Puerto Rico and the Caribbean to enhance the blue economy, which comes from the special grants and special funds that was allotted to Puerto Rico, stemming from Hurricane Maria, to develop not also things on land, but things on water. And blue tide is this mechanism right now with research vessels and everything, pretty much the disposition of the scientific community in Puerto Rico. So I think Adriana has got a picture of my card, my email, the QVR code, and we'd love to have you guys either come to Puerto Rico or more than happy to assist you in this endeavor. Blue Tide, it's at the forefront of the blue economy, which is trying to bring a peaceful union or coexist of the economy, all the water, with the sustainability of the ecosystem. So we are right now, we're working with the underwater, it's called the FADs, FADs, fish acquisition devices. And we're starting to harness all of them. We pretty much setting up along the north coast of Puerto Rico to enhance the habitats for fish and everything. So we'd love to have you guys come on board, right? We'll be more than happy to assist you with what we got. We got a 75-foot vessel, research vessel. It's a floating laboratory. Nothing exists south of Florida. And we have it. We have it right here in Puerto Rico. And we'd love to have you guys contact me, and we'd love to work together. You as a new generation and myself, well, trying to enjoy the ride. Muchas gracias.
Gracias, Edgardo. Un aplauso, a big applause for Edgardo, for Adriana. Super good, because family always make us better. So I invite you all to open your cameras. We take a picture. We have SOA Panama present right here. So we need to protect the corals in Panama as well. So everybody, we give 1 minute. Edgardo, thank you. And blue economy is so important, because this is the language that governments understand. Blue economy.
Very true.
And what's happening with our scuba diver? I'm a scuba diving teacher, and if we don't have corals, then what people will be looking at.
Yes.
And so thank you for that. Hey, everybody's opening the cameras. I love this.
I can tell you quite, when we usually do this with our Caribbean community, we have like 5 people who put on their cameras, and that's it. So don't be surprised if not much people have their cameras on.
Mario, Jonathan, Christopher, let's do it. Let's do it, Joshua. Okay, yes, Christian, excellent. Okay, so what if we say at the count of 3, save our corals? What do you think? Okay, but you have to open your microphone so the corals hear us. Yes. They won't hear and won't help.
No.
Okay, cameras on. Yes, cameras, microphone on. And in the count of 3, we say save our corals. Are you ready? Hey, Katrina, little boy. Hey, Honesty, little boy. Okay, let's do it. Let's do it. So, 1, 2, 3. Save our corals. Hey, guys, thank you for joining. Thank you for joining. This is a Acropora palmata sign. So feel free to give energy to those corals in help, in need. And thank you, Gabi. Thank you, Hannah. Thank you, Adriana, Lourdes, and Khadija, SOA Caribbean, for hosting this. Very important. Let's keep on rolling, Kadi. Maybe some.
Of course. Yeah, well, we already got an invitation for Puerto Rico, and I kid you not when I say we are coming. And like everybody who knows that when I give my word, I give my word. So we will make something happen, and we
Yes.
would get to Puerto Rico next year. Have no fear. I will take all your information from Adiana and see how best I could find a grant and make something happen because it's important. And I want to do it, and I believe that the community present here right now also wants to do it. So why not?
Excellent, excellent.
More than
thank-
welcome.
Eso, thank you, Edgardo, thank you, Edgardo. Emilio, estamos en contacto, mi hermano. Y bueno, gracias a todos. José Camacho, por ahí te veo. Gaby, thank you very much. Hannah, amazing. Adriana, Lourdes, awesome job. Let's save our corals. Save our corals. Save our corals. Save our corals. Save our
And on
corals.
that note, everyone, because if I don't stop, Carlos will keep going. Have a very good night. And thank you all so much once again for joining us. And stay tuned for more things we have coming up. Oh, before you go real quick, on Sunday, we have a next event with Katrina, who's also present on this call. We're going to be talking about deep sea mining, drinking some wine, juice, water, alcohol, whatever you wish, and painting some deep sea creatures as well. So check out our Instagram for more information on that and join us on Sunday. Right. So everyone, once again, thank you so much for joining all our speakers. Like, incredible job. Like, I wish so much more people could have seen this, but it's gonna be on YouTube and everywhere possible. So thank you guys so much for an amazing job and presentation, and Carlos for connecting me with the Puerto Rican family, and Gabby, and for making this possible as well and bringing the vibes and energy as always.
7 countries, 7 countries. We are changing the world. Okay, thank you guys.
All right, bye.
Ciao. Woohoo! Save our
Adios.
corals. Ciao. Save our sharks. Save our
Adios.
ocean. Toda Panama.
Bye.
Arriba. Woohoo! Ciao.
That's it.
Bye. Let me end the meeting.